Hello, I'm Brian Bothner, professor at Montana State University in the Department of Chemistry and Biochemistry. So we are looking at biology as a system versus as individual parts. And so this research takes us from the atomic scale of protein models and virus structure, which you can see here, to the living systems corresponding in cells of metabolites, proteins, and nucleic acids that really come together to make a functioning biological system.
And the research I do primarily focuses on looking at the biophysical nature of proteins in different conditions. And the way we look at this is by using an instrument called a mass spectrometer, and I use a technique called hydrogen deuterium exchange, which I then couple to the mass spectrometer, which then allows us to look at the structure and dynamics of the protein in different conditions. And this allows us to tell about ligand binding sites, mechanism, and all sorts of other information that let us really understand the structure-function relationship of different proteins.
My research focuses on taking a systems biology approach to answer a biological question. And what I'm interested in is looking at how arsenic affects the bacteria that live inside our gastrointestinal tract. So arsenic is something that's relatively common as a contaminant in groundwater. And so there are a lot of people all over the world that are exposed to high levels of arsenic. So what I do is I look at the genes and the metabolites that are upregulated when certain types of bacteria live inside our GI tract. And so how I do this is I use mass spectrometry here in the facility at the biochemistry department, and I look at all these metabolites that are upregulated in response to arsenic. And what I do with those is I take those and I compare them to the genes that are also upregulated or downregulated. And I look at what pathways or what metabolisms are being turned on and turned off that can affect the host health. And I've had a really great opportunity here to learn the entire process of metabolomics here in the chemistry department. So I start at the beginning where I learn how to prep samples and I run them, and then I do the analysis portion as well. So I've learned how to do the entire thing, which has been really beneficial for me. I'm also appointed as a graduate teaching assistant, so I teach a lab twice a week here, and it's one of my favorite things. It's been a really good experience to learn material and then teach it to students and be able to mentor and guide them.